All right, everybody, welcome to the last week of Biology 10 labs. I hope you've had a good semester so far. So we've just got this last little bit of stuff to get through, our final lab, lab number 7, and our final quiz activities. I should say quiz 9 and quiz 10. Let me fix that really quickly. And those are the last couple of activities you'll have for this whole term. So remember to finish up the last bits and pieces by this Sunday at 11:59 because your lab grades are actually finalized on Monday morning and will be sent to your lecture instructors first thing in the AM. So everything has to be done before then. Now, by the end of the week, you'll also notice that all of your lab grades are up to date. So if you do have any questions, comments, or concerns about anything related to lab grades, please do let me know. Just get in touch with me either via email, through a Canvas message, however you guys want to get a hold of me, it's all good. So this week you're going to be looking at what's called the movement of water and water balance. And this whole laboratory, if you go into Canvas, can be found in module 15 for this week. And the lab is titled 7, Diffusion and Water Balance. So we're going to be looking at how the movement of molecules, particularly water molecules, can dictate the way in which life takes place. So once you have downloaded the modified lab manual report that's found in the Canvas modules page, you'll be able to get started with some of the simple and active processes that are associated with the movements of water. Now, when we talk about simple and active processes, a simple process is one that happens naturally on its own. So diffusion and osmosis are two really good examples of that. Whereas there are active processes as well. So I think the best way to consider diffusion is to think about all things as being like a solution. So here on the right-hand side of the screen, you have a little beaker, and in that beaker is a liquid that we call the solvent, and dissolved inside of the solvent are what we call solutes. So every sort of solution has this kind of situation going for it. It has a liquid and something dissolved in it. If you want to get technical, that's what you are as well. You're basically a giant bag of water. Well, 70 to 80% water, depending upon your particular characteristics as a person. But that solute concentration inside of you is also present. So, for example, your blood should be at 0.9% sodium chloride, which is called physiological saline. So that 0.9% physiological saline is what maintains the balance inside of your body. So if you were to consume too much salts, that could throw your osmotic balance off track, which can also lead to high blood pressure. And that blood pressure comes from the water trying to manage the salts. Or if you leave your hands in salt water for too long, they prune up. Or on the opposite side of that, you can have toxicity from too much water. So trying to balance out those things is crucial to cells surviving. So when you get started today, the activity number 1, if you look on the second page of your lab activity, it calls for you to make some hypotheses and to demonstrate a virtual activity for tonicities. So I've gone ahead and navigated directly to that website. So we'll go ahead and we'll switch over to my Internet Explorer screen. And here's the website for this particular activity. So this one, the activity is at glencoe.com/sites/common assets/science virtual labs LSO3/LSO3. So make sure you're getting that off of page 2. And here you'll be able to actually do the virtual osmosis demonstration. So when we first look at these cells, this paramecium from pond water, this Elodea cell that you guys have seen from some aquatic plants, or a drop of human blood, they all have a certain tonicity that's about them. And so when we're looking at this tonicity, we're trying to take and see what happens when we put those cells into different types of solutions. So in nature, an isotonic solution is a solution where you have a net no movement of water. So an isotonic is like happy conditions. So when your blood is at 0.9% salt, that's an isotonic solution. A hypotonic solution is when you have too much solute on the inside of a cell. So the easiest way to remember it is that water always moves towards the solute. So in this case, if we drop the blood cell into this hypotonic solution, the cell itself has more salts in it than the liquid around it. So all the salts are in there, and then all the water is here on the outside. So the water rushes inside the cell. Now, if we go to the opposite side of it and we drop this now bloated cell into the hypertonic solution, so hyper means that there's too much salt in it. So now we can see the cell shriveling up. And that shriveling is also called crenation. So crenation occurs when osmotic balance inside of the blood is perturbed. Now, we can also do that same thing with an LODS cell. So in an isotonic solution, it's happy. In a hypotonic solution, it swells up. But you'll notice that the plant cells have a cell wall on the outside. That cell wall helps to protect them from osmotic damage. Then on the opposite side, salt water shrivels it up. So we still see the cell wall, but the membrane retracts from that cell wall. And then finally, we'll look at our unicellular eukaryotic protozoa. So he's happy, he's bloated, and he's shriveled up. Okay? So go ahead and based on those actions, fill out your table on page 2. So table 1, osmosis and virtual activity. And make sure that you can make your predictions about where the liquids will move and answer the 2 questions there on the bottom. And then on page 2, or I'm sorry, on activity 2, you're looking at how organisms respond to an aquatic environment. So for this one, you'll also be able to look at that LODS cell. So the image on page 3 under activity 2 shows you a leaf and a salt solution. So remember that this LODS cell right here is an actual leaf. Okay, so that is a plant cell. So you'll be able to kind of predict what you think will happen in those 2 solutions and answer a couple of questions at the bottom of page 53. Now, for the last part of your lab, you'll be looking at activity 3, and that's osmosis and solute concentration as an activity. So for this one, go ahead and navigate to the next link that's found underneath activity 3 in your book, and this link will open up an online virtual cell homeostasis lab. So for this one, you'll need to follow through on the directions for this cell homeostasis lab to try to determine how much of a liquid is being diffused inside of what's called the dialysis bag. So in this particular example, you're going to be using an artificial environment, the beaker, and inside of that beaker, you're going to be putting about 1,000 mils of water here, so you guys can see me doing that now. And then you're going to be creating this artificial cell. So dialysis tubing is actually something that they use in hemodialysis for patients that have kidney issues and their kidneys can no longer filter. But basically you'll be seeing how different sugar solutions can allow for more diffusion into and out of this dialysis bag that would actually mimic a standard cell. So you can go ahead and continue through this activity, noticing now that we're putting in different kinds of beakers with different salt concentrations. I'm trying to get ahead of myself here. I'm going to add the water first. And then next we're going to weigh out some, I keep getting too excited. We'll keep filling it up. Anyway, so as we keep adding these things in here, you'll be filling out different weight changes according to the dialysis tubing and what kind of solution that it's actually put in. So when you guys are doing this, you'll see first that we are adding some sugar into our dish here, and we're actually going to add that into the solution. So that'll create a different tonic environment. So you'll repeat that with the other experimental variables, the 10% and the 15% sugars. So go ahead and we'll put this stirring rod, we'll stir it up. So as we go through some of this stuff, make sure that you're able to answer the questions on the bottom of page 56 associated with those sugar solution questions. Okay, so we'll continue on with this, continue making our solutions, and we'll go ahead and add the water into it. Go ahead and we'll take our beaker, put it back. We'll tear that, so we'll zero the weight out. We'll add some sugar. And then we'll go ahead and we'll add the sugar directly into our beaker. We'll stir it up. Man, I'm too quick for it. Okay. And then we'll go ahead and we'll make our third type of treatment here. So after you've followed through all this and you've generated your weight change data from the activity itself, you'll be ready to move on to the fourth activity, which will be effects of temperature. So here we're just looking at solutes, but for that last fourth activity, we'll be looking at the osmotic pressure and temperature effects. So I'm going to go ahead and pause the video and move over to that demo screen next. Okay, so here for this fourth activity, you'll be looking at the actual processes associated with diffusion and temperature change. So you can go ahead and open up the link here, and you can go ahead and hit start, and this will start that diffusion process for you. Oops. There we go, down here at the bottom next, I'm sorry. So go ahead and click that, and it'll go through the process associated with diffusion. And if you continue through this, you'll hit the temperature section, and the temperature section will allow you to answer the questions at the bottom of activity 4. So I want you guys to think about this for a second. What happens if temperature is raised? So think about water when it goes in the microwave. If you increase the free energy or the heat inside of a reaction, it should increase the rates of reaction of diffusion. All right, so you see the particles moving around faster inside of there. Now, if we lower the temperature and we look below, now we see a slower movement of those molecules, so it should slow down that movement or activity. So the last question asks you about temperature and salts, and do they make a difference? And that's the end of page 57. So please make sure that you guys submit this lab report by the end of the day on Sunday, and that'll be your last activity for the semester, assuming you've done your course survey and your quiz number 9. All right, have a good week, guys.